you all. And nice rainy day out there. It's feels really depressing. I wish it was all snow. I know other people don't necessarily, but I do. uh, I don't know if you're getting any flooding in New York, but I know here we're um, maybe seeing some flooding issues. All right, looks like everybody is getting on. Okay. All right. Well, I just want to get the ball rolling here. We have a packed agenda today, and we're going to go a little bit longer than we have because we have three speakers that are joining us today from Cornell, and this team has been working on integrated management for seed corn maggot in New York, and we want to hear everything that they've been up to. And I'm sure it's not easy to cover in an hour and a half, but it's a little more time than we've had, and I hope everybody can stay on. And then I believe, do we have another? Nope, that was Friday. Okay, never mind. Moving on. We're on December 18th, not December 15th. Time it just goes by so quickly. So again, this is continuation of our neonicotinoid webinar series brought to you by the University of Vermont Northwest Extension Crops and Soils Program. I'm Heather Darby from UVM Extension, and I've been leading the program here, but certainly not the only one working on this here in Vermont. And I also want to thank the USDA Crop Protection and Pest Management Program that is supporting our work in this area, and the EPA through a pesticide environmental stewardship grant as well. So just a reminder, we have 2 webinars left in the series. We will be joined by Reed Johnson from Ohio State University on Wednesday, and he'll be talking about reducing bee exposure to corn seed treatment dust, which will be a very interesting topic for us to hear about. And then on Thursday, Steve Donnell from the Ag Innovation Board, again, we already heard from him once, but joined by myself to really get some feedback, answer additional questions, and really try to hear from the community that we've been holding these webinars for, so we can get some feedback on, suggestions and additional questions people would like answered as we move into continued investigation of how we'll be managing neonicotinoids in the future. So we are offering CCA continuing education credits as part of this program. And at the end of the webinar, we'll put the QR code up so people can scan that. And then if you need pesticide applicator credits today, please email Susan Brulette or put your information in the chat so that you can get those credits. All right, so back to our intro slide. So as I mentioned today, we are joined by the Cornell University pest management team, and the group of individuals with us today have been working diligently on developing and maybe further developing integrated management strategies for seed corn maggot in New York. And I think a lot of people have heard about new legislation in New York, and maybe you folks are going to touch on that a little bit, but there definitely is a changing tide in New York in managing neonicotinoids. And so Cornell pest management team is trying to develop recommendations and strategies for farmers to be able to manage seed corn maggot without the use of neonicotinoids in the future. So again, today we're joined by Ken Wise, who's a senior extension associate with New York Integrated Pest Management Program at Cornell. He provides a lot of leadership to applied research and innovative educational programs in IPM for organic and conventional dairies, livestock, and field crop producers in New York. And I know Ken is working with members of our team on other projects related to dung beetles and parasite management. And Dr. Alejandro Calixto is the director of the New York State Integrated Pest Management Program. And he also leads a team of entomologists, educators, and extension specialists in pioneering ecologically resilient pest management practices that lead to safer homes, farms, schools, and communities. And then finally, we're joined by, but not least, Dr. Katja Poveda, who's an associate professor in the Department of Entomology at Cornell University. And her research focuses on the ecology of plant-insect interactions in agricultural systems and their interface with natural systems. And I want to thank all of you for joining us today. It's a real honor, and I know we'll benefit greatly from everything that you're doing and you've learned. And so with that, Alejandro, I will turn it over to you.
Thank you, Heather, and thank you, Susan, for the invitation and the introduction. I'm going to share my screen.
Yeah, and Alejandro, I think as you folks are moving from speaker to speaker, I'll just let you kind of do that. I won't interrupt. And if we want to take a question in between the transition, we can definitely do that too.
Thank you. And
right.
do you guys see the screen, the PowerPoint?
Yes. Yeah,
Okay,
it looks great.
good.
Beautiful
Yeah,
picture of a beautiful fly.
That's a seed core maggot, the pest of the day.
Yes.
Yeah, thank you again. And yeah, Heather, you were mentioning the weather. So this is my fourth winter in the Northeast, and yeah, these gray, dark, and rainy days are pretty tough. So hopefully we'll get some snow, and we'll get the sunshine as well. So again, I'm Alejandro Calixto. I have the honor of serving as the director of the New York State IPM program. We're part of Cornell University. And I just want to give a presentation, more like an introduction to the presentation that Dr. Katja Poveda and Ken Wise are going to give about the different projects that we are developing in New York State towards safer and sustainable alternatives to neonicotinoid insecticides. Just a little bit of background about neonicotinoid insecticides. I'm pretty sure you've heard that on another presentations. They're a group of insecticides that were introduced in the 90s. I always use the IRAC classification as a reference. So the IRAC classification, the Insecticide Resistant Action Committee, they're the ones in charge of classifying the mode of actions of insecticides. So as of now, we have about 36 mode of actions that have been classified, and neonicotinoid insecticides are number 4. Just to give you, it's kind of a reference. Those are products that are relatively old. They have been in the market for about 30 years. They are systemic. They're absorbed by the plant. They move in the xylem and the phloem, and it redistributes in the plant tissue. They are also highly soluble. They move really well in the water. And neonicotinoid insecticides is the most widely used insecticide around the world. It has played an important role against important insect pests in different crops in the past few years. So why are neonicotinoid treated seeds controversial? This is something that is new. There's been a lot of information, particularly started about the 2010, 2012, a lot of work that was conducted in the Midwest, and basically kind of exposing not only the risks, but also highlighting the benefits. And what I want to say is that the majority of the corn and soybean seeds that are planted in the US, they are treated with this insecticide. In the Northeast, it's used mostly as a preventative treatment against seed core maggot, wireworms, and white grubs. The usage, as I mentioned, it's been a lot of more than 10 years that there's been research looking at the risk. And basically, it's becoming more controversial because we know more about these insecticides. There's limited evidence of the economic benefits. There is the problem on environmental risk to non-target species. There's also concern about viable alternatives, and possibly the diamides, which is another insecticide, is a newer mode of action, could be a possible alternative. So In 2020, Cornell released a state-funded report that provided a comprehensive risk-benefit analysis of neonicotinoid insecticides in New York State in agronomic and specialty crops. I have the link and the QR code for that report if you're interested in learning more about that study. And basically, that study focused on where there are risks to pollinators, the economic benefits of neonicotinoid insecticides, and when there are viable replacements. So the key findings of this report, it really exposed a lot of issues related to field crops. So the benefits only exist, they found that benefits of neonicotinoid treatment, seed treatments, only benefits a small portion of the fields that were studied. But of course, that small proportion, there is a benefit that is observed out of the seed treatments. The risk in and near neonicotinoid seed-treated corn and soybean fields is real. So that's where it really exposed some of the problems that we have, and there's different pathways of exposure to neonicotinoids. One is the dusting, and I see that Heather and Susan have a presentation about managing the dust when they're planting seeds. There's also long-term contamination in the soil. So some of these actives, they can persist in the soil. They're up because they're systemic. They're taken as well by plants around those fields, including weeds, and also is leaching into the water. So this is kind of a high level slide where it shows the clearest results. So they look at all the studies they have conducted in the US and other parts of the world around neonicotinoids. And basically what they found is that field crops, more than 96 studies, they found a negative impact of the seed treatments. There's also not only the study that was released by Cornell, but you can also find EPA did a comprehensive study as well, and it's published in their website, where they look at the benefits of neonicotinoid seed treatments to soybean production. And basically what they found is they found no benefits of the seed treatments in soybean production. That's also a very interesting, there's a few publications in that website that I would encourage you to get more familiar with. And most recently, the EPA released this study just looking at the impact of some neonicotinoid insecticides on endangered species. And basically they found, this assessment found that neonicotinoids are likely pushing 200 endangered species to extinction. So again, as I mentioned, there is only limited alternatives to neonicotinoid insecticides in field crops, mainly in corn and soybean. What it was found in that report is that likely the anthranilic diamides are a chemical viable alternative. Some of those are already commercially available. Some corn and soybean is already treated with diamides, but also kind of another thing that was really concerning for us is that there were limited options, limited alternatives, and there is a need of broader development and adoption of IPM alternatives, non-chemical and chemical, in the state of New York. So because all this amount of information that it was presented, there was a bill that it was introduced in the state, and basically the bill recommends the restriction of neonicotinoid insecticides in the state of New York. It's called the Birds and Bees Protection Act. It passed the legislature. It is now sitting in the governor's office, and the deadline for signing or veto this bill is December 23rd, which is just a few days from now. There is the link of that bill as well, the QR code. I would encourage you to get familiar with that bill as well. We don't know what's going to happen if it's signed. How is that going to impact on other states? We wouldn't be surprised in some other states they try to follow a similar approach in the future. So if the bill is signed, it will restrict the sale, use, and distribution of corn, soybean, or wheat seeds that are treated with neonicotinoid insecticides. So these are the main chemicals there's different type of products under neonicotinoids, and the ones that are going to be impacted are these 5 that are listed in the PowerPoint. And that restriction, if signed, will start in January 1st of 2027. The other part of the bill that is going to impact agricultural systems, or well, not so much agricultural systems, but basically the general public, is that it will restrict the application in outdoor ornamental plants and turf, except for the production of agricultural commodities of ornamental plants and turf. And that will go effective on July 1st of 2025. So as of now, the bill says that the New York State Commissioner of Agriculture, we have the authority to suspend the provision of the bill about seed treatments if, only if, there is insufficient amount of commercially available seeds to adequately supply the agricultural market that has not been treated with neonicotinoids. Another important part of that bill is that it instructs the Department of Environmental Conservation, in consultation with the Department of Ag and Markets and the land-grant university system, which is Cornell University and the New York State IPM program, to conduct studies to identify practical and feasible alternatives to neonicotinoids. Those results and recommendations should be submitted to the governor by January 1st of 2026. So without that bill being signed yet, we decided just to take a quick approach and get around and put together a group of people and start working towards developing sustainable alternatives of neonicotinoid insecticides. That funding is coming from our program, and it's basically targeting field crops, vegetables, and turfgrass. In vegetable crops, we have, as of now, 6 projects looking at cabbage, onion, potatoes, snap beans, sweet corn, and squash, and organic vegetable production. And those projects are led by Dr. Van Noll, who is in the Department of Entomology, and Abby Seaman and Marion Souffle, who are a part of our program. We have one project in turfgrass that is being led by Dr. Cal Wickens at the Department of Entomology as well. And then we have up to 6 projects, and we're probably going to expand to more, 2 or 3 more next year. And those are mainly in field crops, in corn and soybean. And you're going to hear more about those projects with Dr. Katja Poveda and Ken Weiss. So these projects in seed treatments in field crops have been around seed-core maggot. So seed-core maggot, as I mentioned, is one of the key pests that those neonicotinoid insecticides are being used against too. These flies over winter pupae in the soil. Early in the spring, they become really active, and they start laying eggs in the soil, and they're attracted to the canned organic matter. When the eggs hatch, they start looking for food, and they seem to be highly attracted to legumes. And of course, we have large amounts of acres that are being cultivated, planted with corn and soybean. So some of those seeds might be impacted by seed-core maggot. So our main focus on the projects that we have in New York State are around seed-core maggot. So this is the group of people that is working in these projects. We have people working in entomology department. We have people working in plant soil sciences and crop and soil sciences. We have people from our regional teams, extension teams, and of course, people from the IPM program involved in these projects. So these are the five projects that you will be hearing today. One is basically trying to go back to the basics of IPM. Just, of course, we know the pest, but we want to develop some tools that are reliable to monitor and help predict that pest. We have some treatments using conventional chemical alternatives, but also we're looking at non-chemical alternatives as using a methyl jasmonate to induce resistance to seed-core maggot. But also we're looking at the impact of cover cropping in seed-core maggot. So we're trying to look at the different aspects, the cultural practices, looking at chemical tools, but also trying to develop a good method to monitor that pest, but also to predict that pest. We have also spent a lot of time doing extension and outreach. There's a lot of seed treatments. It's a relatively gray area. The growers, they don't really know exactly what comes in those seeds, but also they need to understand that we have their back. So we have all these projects that we're developing, all these tools. So we've been very active starting this year, just trying to participate in different grower meetings and trying to deliver the message about these different tools that we're developing. We have also launched a website that is dedicated to these projects, where we have more information about the projects in field crops and vegetables and turf grass. So here's the QR code as well. So if you're interested to visiting that website and learning more about these projects, and we're going to continue updating that website as we continue producing results from our projects. Again, so the public engagement has been really important, and the plan is to continue doing that next year and even expanding to another crops, even though they're not going to be impacted by the bill if signed. We want to make sure that the growers are aware about the ongoing projects that Cornell University is doing to address that issue. And again, this is kind of just to wrap up this introduction. So basically, we are entering the third year. There's going to be an expansion on the trapping systems that we're developing, expansion of the risk tool that is also being developed by Dr. Katja Poveda and one person from our program, Dan Olmsted, some field testing of that methyl jasmonate, which is a novel technology for protecting the seeds. And also, we just hired recently a postdoctoral research economist, and he's going to start doing the economic analysis of the benefit of adopting these tools. That's going to be very important for us once we start pushing for adoption with the growers. We also have an insecticide survey that we launched early this year, trying to get an understanding of the use of these produce in different commodities. That's going to be also information is going to be processed and presented to the public and the state at the end of 2024. And as I mentioned, the bill, if signed, is instructing us to draft an action plan and make some recommendations to the state. And the idea is to have that completed by the end of 2025. And we will continue updating you guys and growers and other stakeholders about the findings of these projects and the recommendations that we will be making to the state. So that's kind of a high-level summary and introduction to the work, the amazing work that is being conducted by Dr. Katja Poveda and her lab, by Ken Wise, the regional teams, and another faculty in Cornell University. With that, I'll be happy to answer any questions you might have.
All right, if you have any questions for Ken, if folks can put those into the chat. I'm not sure if you mentioned, Alejandro, what is the timeline for the regulations, and how does that look in New York?
So again, so the bill is on the governor's office, and she needs to either sign or veto the bill on the 23rd is Saturday, I believe. So if signed, those are the two dates that I have in the PowerPoint. January 1st of 2027 is when the ban, the restriction on seed treatments goes effective. And July 1st of 2025 is when the restriction in outdoor ornamental and turf will go effective. Now, if it's veto, we don't know what's going to happen. I would imagine that it will go back to the Senate, and it will. I would imagine it won't go away. So there's a lot of conversations going on right now in Albany, and but again, I think there is a lot of movement behind scenes around this bill. But those are the two critical dates of that bill. Now, we also don't know if at some point there's going to be another bill that is going to try to expand to another crops. Vegetables was not in the early stages of the bill. Vegetables and fruits were included, and on the last amendment they removed vegetables and fruits. So there is a possibility that in the future that might impact other commodities as well.
Okay. Thanks for reiterating that. And I think there's another question about in the New York, in that risk benefit analysis, 10% of the fields benefited from neonic use. Was there anything in common about those
Yeah,
fields?
that's a really good question. So some it have to do, of course, with the window of planting. So there is, if they're planting that corn on soybean when there's a high activity of seed corn maggot, there's a high chance, the high probability of having an impact. So that's been one common thing. There might be some history as well of high pressure on those fields. But those are the only two that I recall as of now.
Okay, great, thank you. All right, if there are no other questions for Alejandro, we will turn the floor over to Dr. Poveda and let her start with the next segment.
Perfect. Thank you so much, Heather. I hope you're all seeing the big screen, not my
Yes.
notes. Excellent.
Yep,
Thank
we
you.
can see everything.
So I'm just going to build up from Alejandro's introduction on seed corn maggot. So what we are doing in the lab is predicting the damage risk and identifying tools for better monitoring, but also trying to understand how different soil amendments and local management practices are affecting seed corn maggots. And before we start, I just want to acknowledge that this work was a huge effort by a giant team of people. Both our extension collaborators, but also students in the lab have been instrumental to make this all happen. So as Alejandro was saying, the seed corn maggot, it's a fly, so it's the larvae of the fly that are causing the problem. They have a global distribution. They feed mainly on decaying plant and animal material, and this is going to be important as I go at the end of my talk, because they can become pests of corn, legumes, cucurbits, or other crops, and the seeds of these crops, but they also just like the decaying plant material. So if the seeds are not decaying, they will not attack them, and they do not preferentially eat seeds. There are generalist feeding on decaying plant and animal material. If they do attack the seeds, they will do it before and during germination. And obviously this is the biggest problem because it will cause a stand loss. This can be patchy and it's sporadic. It's really hard to predict. So that's what we're working on, trying to understand when it will become a problem. And the next problem obviously is there are no rescue options. So all the work needs to be on prevention and hopefully also understanding better how this pest works. So there's three main research questions I'm going to be talking about today. So first is, can we predict when and where seed corn maggot will emerge and cause stand loss in New York State? Second, how local management practices such as cover cropping exacerbate seed corn maggot infestation. And third, how can we best trap and or monitor seed corn maggot? And I'm going to start with the when and where seed corn maggot will emerge. And I just want to have a disclaimer from the beginning. All our data that we're presenting now are preliminary. We're still collecting data. We are starting data analysis. I will show you what we have and the understanding we have at the moment, but things might change slightly as we analyze more data and have a better understanding of other factors that could be influencing what we have. Okay, so why do we care about when the seed corn maggot can emerge? So if we can predict accurately when the insect emergence is, we could use this for effective management, such as deciding which is the planting time, but also when should we start monitoring a pest if we want to do some type of monitoring. We know that here in temperate regions, each insect will require specific heat accumulation to progress through different life stages. And in the case of seed core maggot here, I'm going to just change this to a pointer, we know that the temperatures, like the range of temperatures in which the seed core maggot can develop is between 39 and 84 degrees Fahrenheit. So we know that some point in April, we reached those temperatures here in New York State, and that's when the seed core maggot hopefully, or is predicted to be emerging when they approach a certain threshold. And we use the degree day values to estimate that heat accumulation. So basically each day that's above 39 degrees Fahrenheit will add to the timing for the pupae to be able to emerge as adults, which is that first adult emergence that we want to predict. And what we want to do is actually upgrade degree day models for New York State. And this is a work that we have been doing with Dan Olmsted, Paola Olaya, former postdoc, and Chloe, a current PhD student in the lab. So when we started this project last year in 2022, the first model prediction was giving us an emergence state of adults between May 2nd and May 16th. So we started sampling last year on April 19, which is 4 weeks before the predicted emergence states, and we started sampling at 25 sites. So just to give you an idea of the sampling, so all the sites here were sampled last year, but the 25 sites that we could use for that early sampling were actually, we got stakes into the ground, are the ones highlighted. So for each site, we had 2 pairs of sticky cards, a yellow card, a blue card, and this was repeated again 100 meters away. And this also allowed us to figure out if the blue or the yellow sticky card is better at catching the adult seed core maggot. So when we started sampling in these 25 farms between April 19 and 22nd, we found adults at all sites during that week. So we knew already that the model was at least 4 weeks off, because on this date, we had adults present at all sites. So it was clear that we started sampling too late in 2022, but at least we could update the model for 3 to 4 weeks from the initial model. So in 2023, which was the year at the beginning of this year, we managed to start sampling 4 sites, so 3 here and 1 here, at the beginning of March. And this was much earlier than we would predict, but I was really paranoid that we will not catch that first emergence. So we put out those first traps the first week of March, and it paid off since we had the first emergence between the 3rd and the 4th week at 3 of those 4 sites, and the other emergence happened a week later in the first week of April. So we know already that our model, the initial model, was definitely off almost by 2 months. And by updating our degree day model, actually the threshold is much lower than it was predicted before. And it's much lower when it's used in many other sites here in the US. Normally they work with a 360 degree day threshold, and we are finding a 67 degree day threshold. We will repeat this experiment next year just to make sure that this is accurate and that we can replicate this. What is important is that we're basically seeing seed core maggot come out before any corn is planted, at least 2 weeks before. So there is no window. That first generation is happening before corn is even in the ground. And I think that was really important to find out. So what we know until now is seed core maggot is emerging already in March in New York State. We will be testing the model accuracy for 2024. We want to include damage risk. So here we are sampling adult emergence and adult abundance over the whole season. We did sample over the whole season. But what we want to do is also to be able to predict actual damage on the seeds, which is what people care about. So to do that, in 2023, we started sample with wire mesh traps. And what this looks like is basically like a wire mesh. So we have a wire mesh, it is just folded. 10 seeds will be placed on the wire mesh, and then the trap will go in the ground. We had wire mesh traps with corn, which is our main interest, obviously, but we also had wire mesh traps with lima beans because we know they're very attracted for seed corn maggot. So if we didn't find any larvae on corn, at least we hope to find some larvae on the lima beans. That's as a bioindicator of the presence of the larvae in the soil. We also use this wire mesh, other sets of wire mesh to estimate germination as a measure of stand loss. So we could count from the 10 seeds we put in the ground, how many actually did germinate. And then we have records of local management practices for all the sites where we're doing the samples. And this will allow us to also know if a certain management, such as cover cropping, for example, or tilling, is affecting the amount of seed corn maggot we're seeing. So just to show you some very preliminary results, if adult presence is predicting seed damage, we actually are not seeing any correlation. So you can see this is the abundance of seed corn maggot adults on the sticky cards. And there's a high variability. These sticky cards are covered in flies, and there is no relationship whatsoever with the amount of seed corn maggot we find in the ground on the seeds. And if we look at the seeds germinated, the germination rates, it also is completely unrelated. So adult abundance is completely unrelated with seed corn maggot present and seed germination. And so we want to, that's why I'm saying we want to update the model and start doing a model on seed corn maggot larvae. So the actual maggots in the ground and the damage that they are causing to the seeds, and see if we can predict that. So right now, we're also establishing the number of generations per season that we're having this last. So 2023, we sampled from emergence until Thanksgiving, and we are continue sampling at one site since the seed corn maggot still seems to be showing up. Like we caught one last week and it was frozen. So we're trying to understand what they do over the winter too. We want to see which generation is actually affecting the timing of planting. And again, we want to see if we can predict maybe the abundance of the previous week of adults will affect the damage next week. And that's something that Chloe, the PhD student, is working on. And again, can we predict planting dates that will minimize the risk? Is there like a low in between the generations that we could use to minimize the risk? But we also want to understand like how local management is affecting it. And I will go into that in a second. So our main goal is to create an interactive tool based on weather, land use around the farm, local management to predict the risk of seed corn damage emergence, but also damage across the state. And we will continue doing that next year. So the next, I don't know, I see a lot of movement in the chat, but I have no idea if it has anything to do with this. No, perfect, good. So the next question I want to address is if local management practices exacerbate seed corn maggot infestation. And if you have looked into seed corn maggot damage, you will find, give me a second, it's not moving, that in the literature, there is a lot of information that the pest is closely associated with organic matter, such as manure and plant residues, that females seek fields with high organic matter to lay eggs on. So there is this information that we're finding everywhere, and we wanted to test if this is actually true in a replicated experiment. Like many of these claims are based on visual observations, but not really a replicated experiment. So we actually wanted to do an experiment to test that. And this is work by my PhD student, Lily Elliott Vidaroy. And so this last year in 2023, in April, from April to May, which is the time that we would be planting corn here in the state, she set up experiments to test how liquid dairy manure, silage alfalfa, as a form of incorporating organic matter into the soil, the mixture between liquid dairy manure and silage alfalfa, and just a regular NPK fertilizer is affecting the damage by seed corn maggot. So each of these treatments was replicated 10 times. And in each of the plots we had wire mesh traps with corn seeds and also lima bean seeds, again, just as a measurement in case we don't have any maggots in the corn. We will always have the lima bean as a very good bioindicator if there are any larvae in the soil. So Lily put together this really nice diagram of what we would expect. These are our expected results. So we would expect that the NPK fertilizer had less maggots in a plot than manure, alfalfa, and the mixture. And we were actually expecting that probably the mixture would be the highest because we have the most amount of organic matter. But what we actually found is exactly the opposite, which was very surprising. This is a non-significant effect, even with our 10 replicates. The amount of variation in the amount of seed corn maggot we can find in a plot is really high, and that is causing this to be not significant. But as you can see, the presence of organic matter as manure or alfalfa does not increase the damage by seed corn maggot. And that's what our data are showing us right now. So at this moment, we found no evidence that the addition of manure or silage alfalfa increases seed corn maggot damage in corn or beans. We see that the importance of manure as a local management practice, we will actually analyze the importance of manure as a local management practice for the risk assessment data. So as I mentioned, we have data for these 50-something farms in 2023, and we had a questionnaire that went into all this management practices. So we know exactly how much damage we find in the field, the abundance of the adults, and we will also know the local management practices there. So we could see, for example, if manure is exacerbating the problem in working fields or not, and compare that with our replicated experiment. And we will replicate this again. So Lily will replicate this experiment again in 2024, and we will modify some of the treatments. Okay, so another local management practice that is really important is cover cropping. And this is a project, or this is a question that Anna Di Paola, a PhD student, actually in ecology and evolution, started working with. And so she is looking at the effect of cover crops and seed treatment on seed corn maggot. And there's two main goals of her project. One is to determine how cover crops influence seed corn maggot damage on the seeds, but also if these cover crops are interacting with seed treatments to influence seed corn maggot damage, and how that's affecting stand loss and crop yields at the end. So with help, again, of our extension collaborators, she detected 16 different farms that you can see here. 10 of those farms had a rye cover crop, and 6 of those are wheat cover crop. And here you can see that we're talking with the growers, she found sites or could convince them to plant one part and not plant the other with cover crops, so that she would have this type of an arrangement where one section of the field had cover crops, one section of the field had no cover crops. And then superimposed on these cover crop sections, she had the 3 different seed managements. So one is neonicotinoids with fungicides, diamides with fungicides, or fungicide only. And this is replicated in the non-cover crop section and the cover crop section. To see how much damage there is because of seed corn maggot, at the moment of planting, she added the wire mesh with the lima bean traps. And 2 weeks after planting, she removed the lima bean traps from the soil. But in addition, she collected 20 corn seeds on either side of the trap to see how much damage there was on the corn seed that was planted in the field. So these wire mesh traps were brought back into the lab where maggots were counted in terms of larvae or pupae. Also, other herbivores were detected, such as wireworms. The proportion of seeds damage was counted, and then here, these are two fellow PhD students from Anna that are helping her collect the yield data. So again, these are preliminary results, but what Anna has been finding until now is that if we look at the cover crop effect and also of the different seed treatment effects on the proportion of seeds damaged for corn seed, there was no significant effect, not of the seed treatments, not of the cover crop, and also not of the interaction. Looking at the same analysis, but on lima beans, it's exactly the same. There is no significant effect of the cover crop effect, of the seed treatment, or the interaction. None of those were significant. When just looking at the effect of cover crops and non-cover crops, which was the main motivation to do the study, again, there is no significant difference between the two, and that's true for corn seed and lima beans. And the tendency is actually the opposite, that the cover crops have slightly less, it's not significantly different, but slightly less amount of damage than the non-cover crops. Anna went a step further and separated the wheat cover crops from the rye cover crops and found that in wheat cover crops, she couldn't find any maggots. We don't know exactly why between cover crop and non-cover crop. And for rye cover crop, again, there was no significant difference. The only significant difference she found was in lima bean rye cover crops, where having no cover crop, actually the amount of seed corn maggot was higher than having cover crops. Again, Anna is still working on data analysis. We think that by including other factors, we can decrease variation, and maybe some of the other results will become significant, but this is what we have right now. So from our current results, we can say that using cover crops does not seem to increase seed corn maggot pressure in field corn, that dry cover crops may reduce seed corn maggot pressure at some farms, and that insecticide and fungicide-only seed treatments perform similarly to each other, even in the presence of cover crops. So there is no interaction there. I want to highlight that this was done on 16 working farms. It's not an experiment as such. It is where growers planted the seeds, and Anna worked with the growers directly. They made all the management decisions, but we also know that these farms have a long history of neonicotinoid use, so there could be a halo effect where there's still neonicotinoids in the ground, and we don't see the effects as strongly as they were if these neonicotinoids were not used over a long time. And the same fields will be hosting an identical experiment in 2024, so we are waiting for those results to see what we'll find next year. So just very briefly, I want to go to our third question, is how can we best trap and monitor seed corn maggot? So from the field sampling in 2022, I showed you that we were using blue and yellow sticky cards. The data are very, very clear, no matter where we did, like which farm we looked at. The amount of flies caught on blue sticky cards will always be higher than on yellow sticky cards. And this is why we're just using blue sticky cards now. From now on, it's normally double as many flies caught on the blue sticky cards. So as a monitoring tool, the blue sticky cards are definitely catching more flies. The other part that we have found that is very interesting is that, as I mentioned, we have been using lima beans in all the field experiments because no matter what we do, lima beans are extremely attractive. In field experiments, in laboratory experiments, they're consistently much more attractive than corn. And so what Lily is starting to do now is to see if we can use lima beans and other volatiles as an attractant for seed corn maggot in traps. Can we develop traps for seed corn maggot using this lima bean attraction? And we know it's attractive for the females to oviposit, and it seems to be also be attractive for the maggots. Like once they're in the soil, that they're more attractive to the lima beans. And Lily is working on that to explore this further. So currently we're looking at the abundance of seed corn maggot on sticky cards, and this would take us months. This is Shay, our key technician who has been identifying flies now for a year. We are working with a professor in agritech to look at artificial intelligence technology and see if we can actually identify the seed corn maggot on sticky cards with artificial intelligence. If this were to work, hopefully it can be developed into something that anybody with a cell phone could use. But we are at the initial stages of this. We want to look into how can we best predict larval damage and stand loss. So can we use previous adult abundance, but also other biotic and abiotic factors that we're seeing in the field. And this is something that Chloe is working on. We want to improve the models to account for damage risk, the soil types. We have soil samples from all these sites, the management practices based on the questionnaires we have for each of the sites, and the landscape composition, which might be really important. And we're starting to prepare for the 2024 field season, where we want to repeat most of these experiments to see that we have a more robust data set, but we're also adding experiments to see the residues that we'll find in the pollen of corn from the different seed treatments, also from the application of the different manure, and also to look at insecticide resistance of neonicotinoids, but also of the alternative of neonicotinoids in seed corn maggots that we find in the field. And with that, I want to thank mostly the team of people. Thank you for your attention, and I'm happy to take any questions if there are any.
Great. That was great. Thank you. So much good information. So if we have any questions, could you please put them into the chat? I'm not sure if maybe this is a question. I guess it is a question, but one of the practices that's very common with cover crops and corn anyway, especially on our conventional farms, is no-till systems where the cover crop is actually sprayed out. And, I think we have all heard about, this attraction of the seed corn maggot to decomposing organic matter. And so all of the fields that we scouted this year here in Vermont were no-till fields that had cover crops. Some of them had manure, some of them didn't, but it was all, the soil wasn't tilled at all. And so the cover crop was sprayed and it died. But anyway, I was just really curious if that was one of your treatments, because it makes me sort of wonder about that, that smell. I don't know if it's like a scent or some kind of, I think of maggots, right? Disgusting, sorry, maggots, like rotting meat, and like, that's what they're attracted to. So if, we're not creating that kind of environment, would they actually be attracted or not? So anyway, just curious of your thoughts on that.
That is an excellent question, and that's actually a question we're working on. So for till and no-till, we have not done that purposefully. Most of the farms, as far as I know, have tillage. But as I said, we have the questionnaires that go with each of our farms. So if we have enough replicates to look at till and no-till, we will definitely do that. But what attracts the maggots and the adults? What is the smell that makes it attractive? That's something we are actually looking into, like really in the lab with volatiles, and we're starting to do those experiments. It's exactly what is it, like what about that rotting smell is it that makes it attractive? Is it certain kind of bacteria? Like, what is really happening? We don't know yet, but that's hopefully a year from now, we will know more about that.
Yeah, I do think some of the concerns that have been brought up here in Vermont have been around, our significant efforts, both, like education implementation that revolve around conservation and building soil health and, increasing organic matter and adding more organic matter. And then, to hear that, oh, that may actually attract more pests. And then
That's not what we're finding, which is
right.
very interesting. And I
Yeah,
think
exactly.
yeah. So I think what we want to, part of this experiment that we are planning right now, which is the attraction of the adults and the larvae, is to find out if maybe the adults are, so this is my working hypothesis, there's no proof for this at all.
Right.
But the adults are attracted to that organic matter. The larvae will prefer that organic matter over the seeds. So actually having the organic matter might not be a problem because it's attractive, but they will prefer that over the seeds. And again, we are starting to do those experiments to test if that is true, because we are not seeing, as I showed in the data, that where we have more adults, we have more damage. Like, those things are completely unrelated. So I think right now, our working hypothesis is that, yes, they're more attractive to manure, they're more attractive to this decomposing matter, but that's going to provide the food for the maggots, and then they will avoid the seeds.
Yeah. Yeah, this is great. Because these are a lot of the same questions that are coming up here. And if there are any other questions, please put them in the chat, because I think I have one more. And I think the other piece that I feel like hasn't really been evaluated, maybe you folks are working on this, but we did hear from John Tucker last Friday, about the impact that these treatments have on other biology in the soil. And so part of what I was also wondering was, well, with more conservation, more organic matter, and this push towards like more biology in the soil, are we actually combating seed corn maggot in a very different way? So even if they're there's more predators and more biology that's also acting against them, even with or without seed treatment. But anyway, I just wonder if you have any comments about that.
I think that's true. I don't want to lean too far out, like, we are just gathering the data, but I think that is actually, like, yes, I think that the seed corn maggot, that it will definitely not exacerbate, at least from the data we have, it's not exacerbating seed corn maggot.
Great.
Problems.
Well, that's really exciting. And I just want to reiterate to everybody, as you've already said, it's preliminary and you're going to continue to do the work, and we'll be really excited to hear about updates. And if you have any questions, please get them in the chat, and hopefully we'll have a few minutes at the end to ask any additional questions. But I think we'll go over to Ken next. And thank you so much. Very exciting work. Ken, take it away.
Thank you. So I'm going to talk a little about some of the alternative seed treatments we're testing, and some observations we've kind of made in this research. So like everybody else, we have a lot of people working on this. I'm really dependent on area specialists, and most of you probably know Mike Davis because he's up in Willsboro. And we'll kind of get into all the different locations that we had. Really tough to present all the data because it's like 100 different graphs, but I did summarize it, and we'll look at how these other treatments work against seed corn maggot. So in 2022 is when we started. Just a little note is that because the way the funding came in, we couldn't really get things planted when I wanted. So in 2022, they got planted around the end of May into June. but 2023, they did get planted from late April all the way through May. So, you know, and when there is some philosophies about soil temperature and seed corn maggot damage and the sort. So in first year we had 4 locations. Second year we had 8 locations with 15 trials. So 2 of the areas we had multiple plantings a week apart. And I'll kind of get into that in a minute. And then in this coming year, we're going to have 14 trials of field corn, and we're adding soybeans to that as well. So we're going to be looking at different, some soybeans as well in different locations. Because of a lot of reasons, we decided we'd do bioassays as well here in Highland, at the Highland lab, Cornell lab. And currently underway, we have a temperature trial. And this is a little bit off in that Katja's worked with, Dr. Poveda's worked with us on kind of developing more of a night temperature, day temperature type of situation while we're testing products. And I'll kind of get into that as we go along. But we have quite an array of research that we're doing. So on the field trial side of things, these are the locations that we ran plots, and I'll show you what the plots look like in a minute. But we had 8 locations this last year. For the previous year, we'll probably have one other new location down in Cattaraugus or Allegheny County next year, but the rest will be very similar areas. And definitely we'll have it in Essex and Clinton County up there near you guys, because that's where we have the majority of our trials. So this is what our plots look like, so we have each of the treatments. So one is diamide, one is a neonic, one this year was fungicide, and one is a control. And I like to point out, and because some of this data could be taken incorrectly, in that we put an attractant over each of these rows, which is bone and meat meal, and they attract seed corn maggots. So we can actually test these different seed treatments. So these are not naturally occurring populations.
Ken, I'm sorry to interrupt you, but we're seeing your website. I don't know if you're showing a presentation. We're not seeing that. We're seeing
Yeah,
like the
sorry.
first slide of the presentation, but it's within like the
It looks like a
Cornell box.
PDF.
Yeah, it
or
looks like a
something.
PDF.
I thought it was a new fancy format that you use, Ken, so I didn't see.
Sorry.
No big deal.
We did test this before we started. Shoot. Let me try this again.
Yeah. No worries. Okay, so we see something that says like comments. No comments to the side. And we see a slide.
Close that.
Yeah, it looks like it's in Adobe.
Yes, I don't know why it's doing that. Here.
Okay, now we can see. Yep, that looks.
All right.
So I think if you just hit full screen.
Yeah, I'll hit slideshow. Okay, is that better?
Perfect. Thank you.
Sorry about that. I don't know what happened.
Thank you, Katja, for bringing that up.
Yeah, thank you. Just so you can see I'll kind of go back to here. So these were the different locations we had in this for the different trials across the state. Actually, that picture right there is actually in Shazy up there in northern New York. So this was our design. Again, bone and meat meal was placed over each of the rows, and so we replicate. These are each plot is, I believe they were 20. I'd have to double check and go back, but I can't see some of my presentation. Give me 1 second here. Sorry. There we go. So these are all 20-foot rows. Each treatment was replicated 6 times within that trial. And then we conducted plant population counts. We also dug up one of the rows and looked at every single seed for whether they were infested or not, which is quite the process, I have to be honest. But the bone and meat meal, you place that over the row, it attracts the seed corn maggot. So some of the data that I'm going to present makes it look like there was quite the population, but they're not naturally occurring populations. They were attracted to that because of the bone and meat meal. So we'd do the plant populations, then we would dig up one of the rows randomly, right or left. And so I'm going to just present mostly the pool data because there's a lot of data for each of the different sites. And we found that plant population was pretty much the best indication of testing these products. And so as you can see, the first one's for Tenza with a fungicide. Then we had just straight up fungicide, which each of these had Vibrant Cinco. And then we had the Neonic, the Cruiser Max, and the control. So as you can see, there was about 5% to 9% damage on each of the treatments. The control did have more seed that did not emerge from the soil. And what's interesting, so this is 15 trials across the state. I found it really strange that the fungicide came out as high as it did, because I thought it would be almost similar to the control, but it didn't. And I'll have some bioassay data to share with you on that a little bit. The control was significantly different than each of the other treatments there. Just to give you an idea of which, so these were controls compared to any of the other treatments. I wanted to find an easy way I could present all these different locations. And so the X stands for the control was significantly different than the treatments. And, you know, Easton is there in Washington County, kind of right up against Vermont. I had a pretty high population of seed corn maggot, actually a very high population. It was the highest out of everything. But I do find it interesting is that as you get farther into May, we had less damage. And this is just speculations. I'm just assuming, kind of as Dr. Poveda showed you the degree day models. I'm wondering if the end of April into May, you have maybe higher populations of the seed corn maggot. And that's why we could attract more of them. Because as you get into the third week in May, we had a lot less of the plots that actually showed damage. So that was kind of a quick summary of the 15 trials we had across the state. We also did this in 2022, where we tested the diamide. We also tested spinosad, and we tested the neonics. And as you can see, there was no significant difference between any of those trials or any of those treatments within the plots that we had. We had, I believe, 8 plots in 4 locations. We had 10 plots in 4 locations across the state. And 8 of those were 4 of those were in Shazy at the Minor Institute, and 4 were in Willsboro. And I had Mike Davis plant them 1 week apart just to see if we could get any significant difference. And in 2022, we only had 1 of the plots up in Shazy that actually had any significant difference. So we got started a little late on this one. And because of that is why one of the reasons we did the bioassays, because we really want to know if spinosad will work as a seed corn maggot. As you know, it's a biological product that you can treat seeds with, and we wanted to see that if it would actually control seed corn maggot. And so just to kind of reiterate, 2024, we will have diamide, neonic, we will have spinosad, we will have fungicide by itself, we will have a control with bone and meat meal, and this time we're going to have a control without bone and meat meal to see what kind of the natural population is doing in those plot areas. And these are the different locations. Highland is down here in the mid-Hudson Valley. Willsboro, you guys are probably well aware of. Shazy as well. Cobleskill is there in central New York, as well as Russia, New York. Canandaigua is western New York. and Scott is down around Ithaca, and then one location down in probably Cattaraugus County, that area down in southwest New York. Oops, I made 2 slides. And we also have soybean treated soybean trials as well in the same method we did the corn. And we have Cruiser, Fortenza, we're going to have spinosad, and it's just this other fungicide that they use on soybeans instead will be the only difference. And we'll have the control with a treatment of bone and meat meal without a treatment in the same locations, hopefully. So our bioassays, we got, because of the spinosad, that we really wanted to know what was going on. And I'm going to show 2023 before 2022, which would be the spinosad is 2022. But because of Dr. Poveda and Jennifer Thaler, we were able to establish a colony of seed corn maggots here in the Highland lab, where we could test these different products, where we had a little bit more control on the number of maggots that went in and infested these seeds. So what we do is we put 1 inch of sand below and 1 inch of sand above the seed. We infest each container with 45 maggots. And Hannah Toltz, who is our technician, she does all this for us here at the lab. We'll place 45 maggots, either first or second instar, into these containers, and we can treat each of the different treatments. The thing is we had this grand idea that we would do these trials where we could do all the blocks at once. So we can't produce that many maggots. So we run 1 or 2 blocks at a time, and a block is each treatment with 5 seeds placed in the environmental chamber. And so it takes a little bit of time because we need to do 10 blocks of these to get 50 seeds treated each. But we had some pretty good results so far. So this is the 2023 seed treatment bioassay where we tested the Fortenza, the fungicide by itself, the Cruiser Max, and the control. And to as expected, the control was significantly different than the Fortenza and the Cruiser Max. It was not significantly different than the Cinco or just the Vibrant Cinco fungicide. It is slightly higher, and what I find interesting is when we were doing those field trials and digging up the seeds and looking at about half the seed in those trials, there was an immense amount of the seed that was germinated, either had entry holes or maggots in them, and they were healthy growing plants. And that was the case with all of them, even the control, but more so with the with the fungicide. They were infested, but they were not we're not dead. They were healthy growing plants. As you can see here, the number of entry holes was the control was significantly different than both the Cruiser Max and the Fortenza, as well as the fungicide was significantly different than both of them as well. But there was more healthy plants growing in our bioassay, and actually in the field itself too, on those trials last summer. And so the 2022 trial, bioassay trial, we didn't take as much data. We're kind of just learning how to do this, and so we just tested whether they're infested or not infested. And so all the insecticides seem to work well. The neonic, the spinosad worked well, and the diamide worked well. The control definitely got more entry holes and the sort. But one thing we didn't do in 2022 is we didn't look at, we would just record the number of entry holes and infestation, but we didn't look at the plants themselves. So that's one step we took in 2023. And we're also in 2020, one experiment we're doing right now, I didn't really post it, but I don't really have any data yet, but we're running, so a lot of consultants and growers say they get more damage when they plant in the colder soil. So what we're doing is we're running a bioassay where the night temperature is at 45 degrees and the day temperature is at 65 degrees, kind of a May, kind of an average general May night and day temperature. And we're going to look at the difference in infestations of these different treatments on those as well. So that's what I have. I don't know if there's any questions. Kind of straightforward. The insecticides seem to work. The fungicides by themselves kind of seem to work as well.
All right. Thanks, Ken. Very interesting. We've heard that too, as far as cooler soil temperatures, more issues. I actually thought that is why we would have seen some issues this year, because it was really cold during corn planting. And actually, we had a very cold season in general. And I'm assuming that's because it takes long, the seed is more susceptible longer, right? Because it's not germinating as quickly. Is that
Right.
true, do you think?
That's the idea. You're exactly right. That's what I've heard as well. I do find the corn seems to be able to withstand pressure and still make it out of the ground. So it'll be interesting to see what happens with soybeans. The one thing about soybeans is, you probably know, you can take out a fairly large population of the seed
Yeah.
and still maintain a yield. So
Right. Yep.
that might be an issue we might have to take. We don't do any of our plots to yield, and those we may take to yield this year. So
Yeah, we did. So we did some a replicated study this year. So we did go out on farm, similar to what you folks were doing, and we planted, treated, and untreated seed. The untreated seed still had the fungicide, so it wasn't completely untreated. And then we just went out and counted populations. We didn't see any difference in the population. We saw a lot of issues with population, but we never found damage from seed corn maggot or wireworm. It was mostly bird damage, like birds pulling seeds out of the ground, a little bit of cutworm. But I was pretty surprised, because, as I had mentioned, the populations of flies that we were seeing in many cases was pretty well aligned when we were like around planting time. So we were kind of shocked there. But then we did take one of the trials to harvest, where we planted every week for 6 weeks, treated and untreated seed. We didn't see any issues with populations over that time. And we started as early as we were ready, which maybe wasn't early enough, but it was the first week of May. And then we went into mid-June, and it actually seemed like we had more issues with the later plantings, which was very shocking to us. And again, this super preliminary, we need to do the study again, but I'm not sure, like you said, if it's always cold weather. Seems like there's a lot of factors where we could see issues, but there were no yield differences. Even if we saw a few less plants here or there, we didn't see any difference in yields at all.
it looks like
I
there was a
do-
question there.
Yep, I do see a question. Yep. Can you explain
So
how the fungicide? Yeah.
Actually, to be honest, I cannot explain it. I don't know if fungicides have any kind of insecticidal properties, but I think maybe in that it protects the seed and gives it maybe from other pathogens that might weaken it a little bit. I don't know, but that's something we really need to look into a little bit more in depth. And I think this coming year, I know Katja does. I know Anna did the same kind of study, so it'll be interesting to see, in the future what that is all about. Because I honestly kind of don't really know at the moment. So,
If I can add something to it, we were talking about that the other day, and there might be, when this seeds start decomposing, they might be like there might be fungi on like decomposing on the seeds, and that might be attractive to the seed corn maggot.
Mm.
So this comes back to the whole, what is really attractive to the seed corn maggot? What is happening? Like some people have been testing that a little bit. It's like, is it the bacteria or is it the fungi around that rotting seed that makes it? And maybe that's how the fungicide is effective. It's just preventing those fungi or to decompose the seed. So I think that's again like a working hypothesis. I don't have any evidence for this, but this is something that I think Swayam Jit or Lily might be looking into a little bit more in the lab to understand that better.
Very interesting. Are there any other questions out there? This group is so quiet today. I don't know if everybody's
got a
depressed over the
question.
rain. Oh, great. Okay,
Yeah.
Anna.
Hi,
take it
Ken.
away.
Thank you for your talk. Good to see your data. I guess I had a related fungicide question. You might have mentioned this, was your control treatment, did that have fungicide on it? Or
No,
was that no treatment at all?
no treatment.
Okay,
So
yeah,
that there was the neonic, the diamide, the fungicide, and the
Mhm.
control.
Okay, yeah, interesting. And that got plenty of damage, as we saw.
Yeah.
Yeah, I think what Katja was saying is accurate to what I saw in my experiments, where the fungicide either could be preventing sort of overall decomposition,
Mm.
or it could be changing the attractiveness of seed to the maggot, right? Like how the seed smells, because it's now coated in this different smelling thing than if it has no coating at all. But yeah, okay. Thanks, Ken. Great
Yeah.
talk,
So-
everyone. Good to see you all.
Thanks, Anna. So I'm curious if you think we should include a untreated control. I had not thought of that. I was thinking more from the, I don't know, like what's the feasibility of accessing all these different seeds? And we were trying to get, same varieties treated and untreated, which was already a nightmare in itself. But yeah.
Yeah, I found a group that will do it almost everything the way we want it. So that was kind of nice. Even sometimes when you buy untreated seed, it's treated with some kind of an acid, and that was hard to not get on there. So yeah, if you need a contact on that, I can give you that. So
Yeah, that would be helpful. So yeah, if we can't even get little lots, I think that's part of the challenge for the growers is just like how to manage all this. Are any farmers that you know of going with untreated corn seed yet?
uh,
You seen any movement?
it's hard to. I think it's hard to get seed without it unless you go organic. So, but I know that like Pioneer is putting both diamide and neonics on their seed at the moment. I'm not sure why they're doing that, but because we were trying to go with Pioneer and they wouldn't treat the seed the way we needed it treated. So
And not field corn, but sweet corn, organic sweet corn. I would imagine that they're planting untreated seed, right?
Yeah.
Ken?
yeah, I think they still treat it with some kind of an acid. I don't know why. Maybe it's longevity or something, or
And there are biological treatments on much of the, I would say, sort of like commercial field corn that's purchased that's certified organic. I don't know entirely what it is. It's, some type of biological of some type. But yeah, I haven't back a few years ago we had some farmers that were going with untreated seed when John Tucker came and spoke to our at our no-till cover crop conference, and people were really inspired by his presentation. But then I think there was some unfortunate pest outbreak that year or the year after, and it was just like, how that goes. And so people were just really shy about it after that. Because really that's the only way to access seed is really completely untreated. Otherwise, most seed companies are not making a lot of movement there, I don't think.
well, Heather, I'd have a question for you. Do you see many fields that you would say were damaged economically by seed corn maggot?
Not that, I mean, in my years here, which are now adding up, so 20 years, I've only visited 3 fields that were, I mean, outright pretty much wiped out by wireworm. And that's because of, our sod rotations. I imagine all of those fields had come out of sod. And it was they were spring tilled fields. So I imagine, for whatever reason, I know that wireworms can build up in sod, and maybe there was also seed corn maggot. At the time, we weren't talking about this. So when I went out, I said, oh, it looks like wireworm or seed corn damage. And I could visually see wireworms. But, and those were 2 conventional fields and one organic field. But outside that, I've never visited a failed field, or a poor population field that was attributed to seed corn maggot. A lot of other things, but not that. Anyway.
Yeah, I have a similar experience. I've been in
Yeah.
only a couple fields that I would say one cornfield back 2000, year of 2001, where it was reduced by 10,000. Outside of that, it's, pretty minor, 4 or 5% maybe. So
Yeah. But there's, clearly there's a lot of concern. So, all right. Well, we are at the bottom of the hour. I really thank all of you for taking the time today. For those that need CCA credits, here they are. And again, if you need pesticide applicator credits, please email Susan and she will make sure that you get those. And I'm really excited to work more with all of you, and hopefully I can use you as a resource as we plan our research here, and hopefully it can help build on what you folks are doing too. So thank you again. I hope you all have a wonderful holiday. And
Thank you, Heather.
yeah,
Thank you.
Thank you,
it
Heather.
was really great. I
Later
appreciate
this
it.
week. Thank you. Bye.
All
Thank
right.
you.
Bye, everyone. Happy holidays. And
Happy
hopefully
holidays.
folks
Thank
will
you.
be joining us on Wednesday to hear from Reed and from Reed Johnson about managing dust coming out of corn planters. All right, everyone. Have a great day. Thank you.